Welcome everyone to today's session. I'm excited to see so many of you joining. And hopefully what we will be able to provide you with is a brief view of the topics that we'll be covering, as well as introduce you to the main program coordinator, Dr. Sona Vasudevan, and some of the other participants and partners that we have joining this program to talk about their need for research in using bioinformatics and using data science. So my name is Elia Brodsky, as Sonalik already mentioned. I'm one of the co-founders of Pine Biotech. So I wanted to kind of briefly introduce what we do, why are we organizing this program, and where can you envision yourself applying bioinformatics after completing a program like this. So probably many of you have heard about the genomic revolution, right? And so the genomic revolution started in the 20th century. We're now in the 21st century, so it really started in the past century, but it didn't happen so long ago. And over the past 50 or 60 years, there has been a tremendous growth in the type of sequencing data, interest in the human genome and other type of genomic data, and really a revolution going on using such genomic data in many different aspects of biotechnology, agriculture, and biomedicine. And essentially, what genomics often boils down to is analysis of the genomic code to identify variation. And that variation could lead to these changes in the phenotype. So essentially, the change in the genomic sequence could lead to a change in the way that cells, tissues, and organisms look and function. And those could be diseases, or they could be natural variation, like different color of skin, different length of your eyelashes, or maybe even different height and things like that. So what we think of typically when we consider genomics is this idea of finding bugs in the code, right? So if those of you that are coming from an engineering background may be familiar with coding and data science, having code work perfectly is almost impossible as you're developing that code. And so oftentimes finding that bug can really help you understand what is going wrong with a function or some kind of an algorithm that you're trying to run. Now, where is genomics applied, especially in the area of biomedicine? Genomics is extremely important for understanding drug metabolism. So we understand how different proteins are able to process different types of chemicals. And typically drugs are those chemicals that we want to make sure that they're metabolized properly. You can also think of genomics in application to cancer, so somatic mutations in tumor development, inherited mutations like in sickle cell anemia, where the adaptation to different diseases has led to these phenotypic changes that are now causing some kind of a susceptibility to disease. Rare genetic diseases that could be linked to repetitive elements in our DNA, and also drug resistance. And obviously now, living in this pandemic, we know about genomics so much more because it's been in the news, it's been in the media, everybody knows about the mutations, the variants of concern, everybody has been hearing about this. And so it's an extremely important domain for research, but also an application to different areas in biomedicine. In fact, today, genomics is truly in the clinic. And so you can see here an example of a report that is being produced to kind of establish risk for developing certain types of conditions. And this is a routinely used method in the medical field today, at least in some of the Western countries. And so I think what's very important is that genomics is moving away from just being purely in research and being actively used in the clinical setting. And we see this as a very important development, primarily because the cost of sequencing is much lower, availability of reference data is much greater, and so there is already a clear and established relationship between specific changes in the DNA code and specific diseases and conditions that could be clinically categorized. But it hasn't always been like that, and even today there's an active research also on the impact of specific mutations, because if you consider how these mutations occur and how do they actually lead to diseases, it's a complicated, coordinated kind of an effect of multiple factors. So here you can see the significance of DNA variation in cancer, where acquired mutations, germline mutations, mutations in different types of genes, and in DNA repair genes, all of those come together to form a certain level of risk, after which there could be an event that could trigger some of those important changes in critical genes and ultimately lead to what we know as unregulated tumor growth and development. And so these types of changes are really important for us to understand from the basics of genomics and the basics of what this data could show us about these diseases, because detecting those changes early on and also knowing the context of specific mutations can really help us understand how to treat diseases that previously were essentially untreatable. Now, more than just individual mutations, there are many different types of mutations. So there are many different types of variations, like multiple nucleotide changes at the same time, changes that accumulate in a specific region, maybe translocation, so certain regions even moving to another position, or maybe copy number variation, right? So many different types of changes in the DNA. And importantly, it's not just the human DNA, but also the DNA of many different other bacteria, viruses, and parasites that are important for the disease progression and treatment. And so I hope that in this program, we'll have an opportunity to see how genomic data could be used to understand many of these conditions, and how can we study genomic data so that we understand the important factors that are essentially critical for understanding of disease, treating disease, tracking epidemics like the epidemic that we're seeing now, or other potential epidemics that we might see in the future. But I think the critical part of this program is that it will provide you with an insight about next generation sequencing data, how that data is generated, what does this data really represent in terms of the technical part of genomic data sequencing, and then will allow you to evaluate some of this genomic data using these practical data sets that we will take to understand where do we see just simple variation, where can we track mutation, how do we then categorize mutations, and understand what do these mutations really mean for the phenotype. And so we will make sure that in this program, after you complete the training, you will be able to understand the different terminology of what is a nucleotide, how can we categorize different changes of nucleotides, how do we then annotate those nucleotides, and how can we essentially find this association with a phenotype, including some examples of what these phenotypes might mean. Now, all of this together is really the domain of bioinformatics, because bioinformatics combines informatics, the information technologies that allow us to retrieve, store, annotate, and efficiently use different types of information, statistics, or using statistical methods to be able to analyze such data, and the domain where we apply this in the domain of life sciences or biology. So that includes biological research, biotechnology, agriculture, genetics, and biomedicine. And essentially, because of this growth of data, the availability of data, and the need to use this data in a meaningful way, we have developed this OmicsLogic training that includes different types of data repositories, mentorship, and training. And this program is one example of such training that we offer to be able to equip the next generation of life science researchers, clinicians, and industry professionals that will essentially transform the way medicine is being done, the way that new drugs are being brought to market, and the way that we can innovate early discoveries that will inform different types of applications in the future. So the reason why we call this OmicsLogic is because the data that we are describing here, genomic data, is just one of the omics data types that is becoming more and more useful and prominent in this field. And so OmicsLogic combines different user-friendly tools, including different methods of statistical analysis and machine learning. It includes the logic behind experimental design so that we can understand the technical variation in the data. It gives you access to big data mining tools that you can easily use to study this data. And it provides also examples of how this data could be efficiently analyzed to spot significant changes in the context of real-world studies. And so this OmicsLogic program is one program that is going to be available on June 21st. And it provides you with a complete roadmap to get started and learning about bioinformatics and about genomics in general. So in this program, you'll be able to find data sets, explore them and visualize them, understand research methods, and get insight and understanding, potentially leading you to an independent practice project that you can develop in a short period of time. So to kind of give you a little bit more detail about OmicsLogic and beyond this program that we're discussing today, what it offers, I would just briefly ask all of you to go to our OmicsLogic portal, which is learn.omicslogic.com. And so I am, sorry, posted in the wrong chat here. Okay, thank you very much. So to kind of give you a sense of how this program will work and how all of this content is going to be organized, let me briefly show you how you can get started today. So if you could go with me to our portal, and please let me know in the chat once you are there. Okay, you can put there or yes or something. You will find that there's a button to log in, and you can create a free account on here or log in with your social media. And so I want to show you briefly how this is going to work. Okay, thank you, Vasily, Manali, right? Thank you, Sonanika. So please let me know once you're in and you've registered. Punita? Okay, anyone else? If you already have an account, once you log in and you're in, you can go right here to programs. And on the program page, you will find genomics in the virtual lab. And so this program right here is going to provide you with all of the information about today's session and what you will learn in this program. And after a few minutes, we will hear a little bit more about what are the different topics and how this is going to be delivered. So here you can see an overview of the schedule and some of the important tools that you will get access to. So let me briefly tell you what are some of these program resources that you will be getting access to as a part of this program. So for different parts of the program, we will leverage OmicsLogic resources that are designed to help you learn about genomics. And this includes asynchronous coursework with theoretical material, quizzes, and practical assignments. Our cloud-based analytical server, where you can analyze large genomic data and visualize results. The OmicsLogic Code Playground, where you can practice coding in R and Python, as well as a repository of project examples that will guide you from data, the logic of the analysis, to the practical exercises that you can replicate the analysis that was described in the publication. So first of all, we will have an asynchronous course that you can join, and a lot of this you can actually start looking at for free, which is an introduction to genomics. So if you're absolutely new to genomics, this is the place to start. You can go to this course to understand the applications of genomics, some of the basic terminology, maybe some refreshment of your molecular biology, so you understand what is DNA, how is it translated into proteins, and things like that. And then you will also learn about how genomic data is generated and how you can analyze this data to extract meaningful information from it. So to get started, you actually don't need to know a lot of coding. All you will need to know is the logic behind what you are trying to do. So what is the data described and how you might potentially analyze this data. And so that is going to be done using the t-bioinfo server, which is a big data analysis platform that provides you with easy to use graphical user interface where you just push buttons and get some kind of a result immediately. And so on this platform, you will see these maps, logical maps of complex analytical pipelines that will allow you to process data from raw format to a structured format. And once that analysis is done, you will get introduced to important functions and tools using R and Python that will provide you with a clear insight about how coding is a critical component of bioinformatics and what you can do using these existing packages to quickly get results of sequence analysis and annotation. So again, we hope that this can get you started. And even before this program is launched, you can start learning about the fundamentals of genomics, of code and analysis, and the applications of bioinformatics about these kinds of biomedical applications. So now to talk a little bit more about genomics in the virtual lab, which is an intensive program that is intended to provide some of the basic material and the grounding in genomics, as well as see some of the potential applications of genomics to biomedical challenges, I want to describe how these programs are going to leverage some of these asynchronous resources that I briefly spoke about and give you an opportunity to immerse yourself into this genomic data analysis experience that will combine interactive sessions on Zoom like we're having today, self-guided study materials that you will have access to, and some of these practical assignments that will include in-class assignments, but also assignments that you can do yourself, and then present your results to your peers and to the mentor of the program.